Chapter 2, Human Reproduction of Biology, Class 12. The main topics being the male reproductive system, the female reproductive system, gametogenesis, menstrual cycle, fertilization and implantation, pregnancy and embryonic development, parturition and lactation. As you are aware, humans are sexually reproducing and viviparous. The reproductive events in humans include formation of gametes, gametogenesis, sperms in male and ovum in females, transfer of sperms into the female genital tract, insemination, and fusion of male and female gametes, that is, fertilization leading to formation of zygote. This is followed by formation and development of blastocyst and its attachment to the uterine wall, implantation, embryonic development, gestation, and delivery of the baby, that is, parturition. We have learned that these reproductive events occur after puberty. There are remarkable differences between the reproductive events in male and female. For example, sperm formation continues even in old men, but formation of ovum ceases in the women around the age of 50 years. Let us examine the male and female reproductive systems in the humans. Basically, they are talking about humans are sexually reproducing. Yes, they are viviparous. They do not lay eggs. They do not lay eggs. The reproductive events in the reproductive events listing, the first one is gametogenesis, followed by insemination, followed by fertilization, then is implantation, then the period 9-month period in humans of pregnancy or gestation, then it is the delivery, basically the parturition. And they gave a fact here that the sperm production continues even in old men, basically until the end of a male person's life it will continue, but in females at the age of 50 itself, 50 to 55, the menstrual cycle will cease. Now let's see the first main topic here, the male reproductive system. The male reproductive system is located in the pelvic region. It includes pair of testes along with accessory ducts, glands, and external genitalia. Basically, it's located in the reproductive system is located in the pelvic. It includes testis, a pair of testes, along with accessory ducts, glands, and external genitalia. The testes, let's see the diagram first. Diagrammatic sectional view of the male pelvis showing the reproductive system. This is the ejaculatory duct. This is the urinary bladder. This is the ureter. From kidney, this is the seminal vesicle. Urinary bladder, yes, vast difference. This is the testis. This is the penis. This is the glans penis. Urethra, penis, which is whatever there is penis, that is under external genitalia. And this is the prostate gland. And there should also be a bulbourethral gland, which we see in this in case of this diagrammatic view of male reproductive system, where this is the tip portion is the glans penis. This is the foreskin. This is the testis. This is the urethra here, which is acting as a urinogenital tract. This is the bulbourethral gland or the Cooper's gland. This is the prostate gland. This is the seminal vesicle, urinary bladder, ureter, vast difference, epididymis, vas afferentia, reed testis, testicular lobes. Basically, testis contains seminiferous tubules, reed testis. Reed testis opens into vas afferentia here. Vas afferentia opens into epididymis. Epididymis then leads to the vas deferens. Vas deferens is connected to the ureter, which will together form the ejaculatory duct, and this duct is basically the urethra, and urethra leads to the glans penis, and it will be taken out. Okay, this is the rectum and anus also mentioned here. The testes are situated outside the abdominal cavity within a pouch called a scrotum. The scrotum helps in maintaining the low temperature of the testis, 2 to 2.5 degrees lower than the normal internal body temperature. necessary for spermatogenesis, formation of sperm. In adults, each testis is oval in shape with a length of about 4 to 5 centimeter and a width of about 2 to 3 centimeter. The testis is covered by a dense covering. Each testis has about 250 compartments called testicular lobules. Okay, let's see again. Basically, the testes are points to be remembered. The testis is situated outside the scrotum, outside the abdominal cavity within a pouch called scrotum. As for why, it is to reduce the or maintain the body temperature, maintain the testis temperature to be 2 to 2.5 degrees lower than the normal internal body temperature. Okay, that is second point to be remembered. This is due to net spermatogenesis. The third information is the testis is oval in shape. It is 4 to 5 centimeter in width and 2 to centimeter in 4 to 5 centimeter in length and 2 to 3 centimeter in width. And the testis, and one more important point to be remembered, is the testis is indeed covered by a dense covering. And each testis, another information to remember, each testis has about 250 compartments, and these compartments are called testicular lobules. Each lobule contains 1 to 3 highly coiled seminiferous tubules in which sperms are produced. Each seminiferous tubule is lined on its inside by two types of cells called male germ cells, spermatogonia, and Sertoli cells. The male germ cells undergo meiotic division, finally leading to sperm formation, while Sertoli cells provide nutrition to the germ cells. The regions outside the seminiferous tubules, called interstitial spaces, contain small blood vessels and interstitial cells or Leydig cells. Leydig cells synthesize and secrete testicular hormones called androgens. Other immunologically competent cells are also present. The male sex accessory ducts include reed testis, vas afferentia, vas deferens, epididymis, and vas deferens. Let's read that again. Let's important points remembered. Each lobule contains 1 to 3, 1, 2, or 3 testicular lobules, not testicular lungs, seminiferous tubules, in which these seminiferous tubules are the one that produce the sperm, and in each of these seminiferous tubule, it is lined, this seminiferous tubule is lined by two types of cells. The first type is the male germ cells, which is basically the spermatogonia cells, which are diploid, and the Sertoli cells, which are also diploid. The male germ cells, spermatogonia, undergo meiotic division, reduction division, to leading to sperm formation, whereas Sertoli cells, which give nutrition to the sperm, or germ cells, we can say. The regions, and now coming out of the testis, the regions outside the seminiferous tubule, actually inside the testis itself, the regions outside, not in these compartments, where whatever these testicular lobules, which are 250 compartments, outside these 250 compartments, in the interstitial spaces, there are small blood vessels as well as cells called interstitial cells or Leydig cells. And these Leydig cells synthesize and secrete testicular hormones called androgens. Other immunologically competent cells are also present, that is, in the interstitial spaces. The male sex accessory ducts include reed testis, vas afferentia, epididymis, and vas deferens. The seminiferous tubules of the testis open into the vas afferentia through reed testis. First is seminiferous tubule, followed by reed testis, followed by vas afferentia. The vas afferentia leaves this open into epididymis located along the posterior surface of each testis. A point to be remembered, the vas afferentia leaves the testis and opens into epididymis. And epididymis is located along the posterior surface of each surface. There is the posterior surface, this is also the posterior surface. The epididymis leads to vas deferens that ascends to the abdomen and loops over the urinary bladder. And this epididymis leads to vas deferens that leads to the abdomen and loops over the urinary bladder. It receives a duct from the seminal vesicle and opens into the urethra as the ejaculatory duct. Basically, this vas deferens will receive a duct from a seminal vesicle, and this opens into the urethra as the ejaculatory duct. These ducts store and transport the sperms from the testis to outside through the urethra. The urethra originates from the urinary bladder and extends through the penis to its external opening called urethral meatus. Basically, the points to remember, then the axillary ducts, what is leading to what, and the urethra originates from the urinary bladder and extends through the penis to its external opening called as the urethral meatus. This is the urethral meatus, this is the external opening. This is the sectional view of the mammalian testis, view of the seminiferous tubules. These are the seminiferous tubules. These are the cells, lining cells, which are basically spermatogonia cells. Spermatogonia cells, and there are sperm here, 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 and there are also spermatogonia cells, Sertoli cells, which provide nutrition to the sperms, which we are going to learn further about in the spermatogenesis topic. And let's continue. The penis is the male external genitalia. It is made up of a special tissue that helps in erection of the penis to facilitate insemination. The enlarged end of the penis is called the glans penis and is covered by a loose fold of skin called as the foreskin. The male accessory glands include a paired, basically they described the accessory ducts in the before paragraph. Now they are describing the accessory glands. The first gland is the seminal vesicle, next is the prostate, and a paired bulbourethral gland. Seminal vesicle glands are paired, prostate gland is unpaired, bulbourethral glands are paired. Basically, seminal vesicle, bulbourethral or Cooper's gland are paired, but prostate gland, which is not paired, is unpaired. Secretions of these glands constitute the seminal plasma, which is rich in fructose, calcium, and certain enzymes. Remember, a point to be remembered, seminal plasma, secretions of these glands constitute the seminal plasma. Semen constitutes sperm and seminal plasma. Sperm is produced by the testis, whereas the seminal plasma is produced by the secretions from these glands, and it is made up of fructose, calcium, and certain enzymes. Secretions of bulbourethral glands also help in the lubrication of the penis. Basically, they said they gave the function of the bulbourethral gland, that is going to lubricate the penis. The female reproductive system. The female reproductive system consists of a pair of ovaries along with a pair of oviducts, uterus, cervix, vagina, and the external genitalia located in the pelvic region. Both male and female reproductive systems are located in the pelvic region, and the female reproductive system consists of a pair of ovaries, the main sex gland, or not sex gland, sex organ, and there are oviducts, which we call it as fallopian tubes as well, and there is the uterus, which we call womb, and cervix, the gateway to the uterus, and vagina, the birth canal. External genitalia is the clitoris, and it is located in the pelvic region. These parts of the system, along with a pair of mammary glands, are integrated structurally. Point remembered, these parts of the system, along with a pair of the mammary glands, are integrated structurally and functionally to support the process of ovulation, fertilization, pregnancy, birth, and childcare. And point remembered here is that mammary glands are also included in the reproductive system, and they are integrated structurally and functionally to support the process of ovulation, fertilization, pregnancy, birth, and childcare. Ovaries are the primary female sex organs and that produce the female gamete, the ovum, and several steroid hormones, the ovarian hormones, estrogen, progesterone. The ovaries are located on each side of the lower abdomen. Each ovary is about 2 to 4 centimeter in length and is connected to the pelvic wall and uterus by ligaments. Basically, the testis was 2 to 3 centimeter I think it was 3 to 4 centimeter, I believe. 4 to 5 centimeter in length and 2 to 3 centimeter in width. But the ovaries is 2 to centimeter in length. Each ovary is also covered by a thin epithelium which encloses the ovarian stroma. In the testis, we saw it was covered by a dense membranous tissue. Here it is covered by a thin epithelium which encloses the ovarian stroma. The stroma is divided into two zones, a peripheral cortex and an inner medulla. Is seeming like a adrenal gland. If we see the adrenal gland, it's also divided into the cortex and the medullary region. Here as well, it is the stroma, which is the ovarian stroma, is divided into the two zones of a peripheral cortex and an inner medulla. This is the diagrammatic sectional view of the pelvis showing the reproductive system. This is the uterus, this is the womb, this is the cervix, this is the birth canal vagina, this is the rectum and anus, this is the excretory system. Not excretory, it is digestive actually. Part of the digestive. This is the external genitalia. This is these four are the external genitalia. This is the urethra. Wait, is urethra and pubic symphysis, urinary bladder leading to the urethra here. And there is above the urinary bladder, we see the uterus. Okay. There is a diagrammatic sectional view of female pelvis showing a reproductive system. Oviducts, the fallopian tubes, uterus and vagina constitute the female accessory ducts. The uterus, this is a point to be remembered. Oviducts, which are the fallopian tubes, uterus and vagina constitute the female accessory ducts. Even uterus and vagina constitute the female accessory ducts. Cervix is not included here. Basically, vagina, uterus and the fallopian tube constitute the female accessory ducts. Each fallopian tube is about 10 to 20 centimeter long and extends from the periphery of each ovary to the uterus. The part closer to the ovary is the funnel-shaped infundibulum. The edges of the infundibulum possess finger-like projections called fimbriae, which help in collection of the ovum. After ovulation, basically this is the uterus structure. I mean, not uterus, female reproductive system structure. This is the vagina. 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 This is the cervical canal. This is the cervix, basically this one. This is the main ovary. The walls, the walls of the uterus are again divided into three walls with perimetrium, a thin layer. Myometrium constitutes of the smooth muscles, help in rapid contractions during parturition, and endometrium, basically the secretory layer, basically. And this is the oviduct fallopian tube, which has three parts, which is the infundibulum, a funnel-shaped structure, and consisting of fimbriae, which collect the duct, collect the ovum when ovulation happens, and a broader region of ampulla, followed by isthmus. Basically, the fertilization happens at the ampullary region or the ampullary isthmic junction. And this is the uterine fundus. This is the uterine cavity. This is where the implantation happens. This is the fimbriae, this is the structure. The infundibulum leads to a wider part of the oviduct called the ampulla. The last part of the oviduct, isthmus, has a narrow lumen and it joins to the uterus. Uterus single, and it's also called the womb. The shape of the uterus is like an inverted pear. Point to be remembered. It is the shape of an inverted pear. It is supported by ligaments attached to the pelvic wall. The ovary is also supported by ligaments. The uterus is also supported by the ligaments which are attached to the pelvic wall. The ovaries are supported by the ligaments attached to the uterus, but uterus is supported by the ligaments attached to the ovarian walls. The uterus opens into the vagina through a narrow cervix. The cavity of the cervix is called the cervical canal, which alongside vagina forms the birth canal. The wall of uterus has three-layer tissues: the external thin membranous perimetrium, middle thick layer of smooth muscle, myometrium, and the inner glandular layer, the endometrium, that lines the uterine cavity. The endometrium undergoes cyclical changes during menstrual cycle, which myometrium exhibits strong contraction during the delivery of the baby. Basically, they say the functions of the layers. They didn't give function of the perimetrium. It's basically covering the uterus or the reproductive system. Myometrium is basically a smooth muscle which helps in undergoing rapid contractions during birth or delivery period, and endometrium is a glandular layer. Glandular meaning it can secrete things, it can secrete hormones, and it is what leads to the removal. It forms a lining for the implantation of the basically the zygote or the not zygote embryo after fertilization, and it is what leads to the menstrual cycles. The female external genitalia include, then the next topic is about the external genitalia, and it includes mons pubis. Mons pubis is and labia majora and labia minora, hymen, and a clitoris. Total of 5 things: mons pubis, labia majora, labia minora, hymen, and a clitoris. Mons pubis is a cushion of fatty tissue covered with skin and pubic hair. This is to be remembered. Mons pubis is a cushion of fatty tissue covered by skin and pubic hair. Labia majora are fleshy folds of tissue which extends down from mons pubis and surround the vaginal opening. The labia minora are paired folds of tissues under the labia majora. The opening of the vagina is often covered partially by a membrane called as hymen. The clitoris is a tiny finger-like structure which lies at the upper junction of the two labia minora above the urethral opening. The hymen is often torn during the first coitus. However, it can also be broken by a sudden fall or jolt, insertion of a vaginal tampon, active participation in some sports like horseback riding, cycling, etc. In some women, the hymen persists even after coitus. In fact, the presence or absence of hymen is not a reliable indicator of virginity or sexual intercourse. Basically, the female external genitalia includes mons pubis, labia majora, labia minora, hymen, clitoris. The function, basically what mons pubis is it is a cushion of fatty tissue covered by skin and pubic hair. Labia majora are fleshy folds of the tissue which extend down from mons pubis to surround the vaginal opening, and labia minora are paired folds of tissue under the labia majora. Hymen is a covered partial membrane, and these points are to be remembered. And this diagrammatic sectional view of the mammary gland. Yes, this is the diagrammatic sectional view of the mammary gland. This is the pectoralis major muscle. This is the ribs. This is the muscles between the ribs, intercostal muscles. This is the pectoralis major muscle. This is the fat tissue. The first is the first constituent of the basically the mammary gland is the mammary lobe. Then is the mammary alveolus, mammary duct, ampulla. Both in case of the reproductive system, and here in the mammary glands, both the ampulla are broader regions, and here this ampulla leads to a lactiferous duct followed by an areola and a nipple. First is the fat, second is the mammary lobe to mammary alveolus. This is to be remembered: mammary lobe, mammary alveolus, mammary duct, ampulla, lactiferous duct, areola, and nipple. Mammary lobe, alveolus, Alveoli we see in respiratory system as well. And here also it's a similar structure like a balloon or something like that. Then followed by the mammary duct, which are linking the mammary alveolus, then leading to a broader duct of the ampulla. Then a complete attachment is a lactivorous duct, and a dark region surrounding the nipple is called areola. Finally, the tip called the nipple. A functional mammary gland is characteristic of all female mammals. The mammary gland are paired structures, breasts that contain glandular tissue and variable amount of fat. The glandular tissue of each breast is divided into 15 to 20 mammary lobes containing clusters of cells called alveoli. The cells of alveoli secrete milk. A point remembered, the glandular tissue of each breast is divided into 15 to 20 mammary lobes. We saw that mammary lobes, after that was mammary alveoli, then was ampulla, then was the lactiferous duct, then was the nipple. So the mammary lobes before the mammary alveolus are 15 to 20 in number, and they constitute a cluster of cells called alveoli. And the cells of alveoli, point to remember, cells of alveoli secrete milk, which is stored in cavities lumens of the alveoli. Lumens are open spaces. The alveoli open into mammary tubules. The tubule of each lobe joined to form a mammary duct. Several mammary ducts to form a wider mammary ampulla, which is connected to the lactiferous duct and opens at the nipple, from where milk is expressed during breastfeeding. Gametogenesis. The primary sex organ, the testis in the males and the ovaries in the females, produce gametes. Let's see the diagram first. The diagrammatic sectional view of a seminiferous tubule. We saw that there are 250 compartments called testicular lobules, and each testicular lobule is lined by each testicular lobule, or each compartment has a 1 to 3 in number of seminiferous tubules, and each seminiferous tubule is a seminiferous tubule, and each seminiferous tubule is lined by spermatogonia germ cells. These are the spermatogonia cells, and also contains the Sertoli cells, and these are the primary spermatocytes, secondary spermatocytes, followed by the spermatid, then the spermatozoa, and these are the Sertoli cells which nourish the sperm. Here if we see, it is dipping its head into the Sertoli cells to gain nutrition. Then this is the structure of a sperm. This is the sperm is mainly divided into 3 regions of the head, the body, and the tail region. The 3 regions, and the head contains before that the whole body is covered by a thin plasma membrane, and the head constitutes the acrosome, which helps in penetrating the zona pellucida layer of the ovum, and contains the main genetic material, which is a nucleus containing chromosomal material. The sperm is diploid, and the neck region as well. Then there is the middle piece, which is the body region, which contains numerous mitochondria, which are the energy source for swimming, and tail for vigorous motility. Now let's read gametogenesis. In the testis, in males, and ovaries in the female produce gametes, the sperms and ovum respectively, by the process called gametogenesis. In testis, the immature germ cells spermatogonia produce sperms by spermatogenesis that begins at puberty. The spermatogonia, the singular is spermatogonium, present on the inside wall of the seminiferous tubule, multiply by mitotic division and increase in number. Okay, each spermatogonium is diploid, point to remembered, and contains 46 chromosomes and 23 pairs of chromosomes. Some of the spermatogonia, called spermatocytes, periodically undergo meiosis, and a primary spermatocyte completes the first meiotic division, reduction division, leading to the formation of two equal haploid cells called secondary spermatocytes. Basically, a meiosis division is divided into meiosis 1 and meiosis 2, and some of the special cells and each called each spermatogonia is diploid and contains some of the spermatogonia called primary spermatocytes. These primary spermatocytes periodically in various time intervals undergo meiosis 1 to produce the secondary spermatocytes which are haploid. Then these secondary spermatocytes which have only 23 chromosomes each, the secondary spermatocytes undergo the second meiotic division, which is the meiosis 2, to produce two secondary spermatocytes undergoing meiotic division to produce 4 equal haploid spermatids. What would be the number of chromosomes in the spermatids? It would still be haploid only, because meiosis 1 to 2, the result will be 4 haploid gametes. The spermatids which are formed from the secondary spermatocytes meiotic division, meiosis 2, the spermatids are transformed into the spermatozoa sperms by the process called as the spermiogenesis. Spermatogenesis is the formation of sperms. Spermiogenesis is the transformation of spermatozoa, and is transformation of spermatids into spermatozoa. After spermiogenesis, sperm heads become embedded in the Sertoli cells and are finally released from the seminiferous tubules by the process called as spermiation. Basically, Sertoli cells, we know that they provide nutrition, and these sperm after spermiogenesis, the sperm heads become embedded into the Sertoli cells and for nutrition, for nourishing, and they are finally released from the seminiferous tubules by the process called as spermiation. Spermatogenesis, formation of sperm. Spermiogenesis, transformation of spermatid formed from secondary spermatocytes formed from primary spermatocytes of the spermatogonia male germ cells. And spermiation, release of the embedded sperm. Spermatogenesis starts at the age of puberty due to significant increase in the secretion of the gonadotropin releasing hormone. This is a hypothalamic hormone. The increased level of gonadotropin then acts on the anterior pituitary gland and secretion of 2-gonadotropin, luteinizing hormone, and follicle stimulating hormone FSH. Basically, this puberty starts during the age of puberty. Time of puberty, spermatogenesis starts because there is a significant increase in the gonadotropin release hormone, the GnRH release hormone. Basically, this is released by the hypothalamus, and this gonadotropin release hormone acts on the pituitary gland, and pituitary gland secretes the luteinizing and the follicular stimulating hormones, these LH and FSH hormones. And how they act is, LH acts on the Leydig cells and stimulates synthesis and secretion of androgens. Point to remember, LH, luteinizing hormone, acts on the Leydig cells, which are the interstitial cells present in the interstitial spaces of the testicular lobules containing blood vessels. They secrete androgens, and this LH acts at the Leydig cells, LH, LC, luteinizing hormone and Leydig cells, and stimulates synthesis and secretion of androgens, stimulating the interstitial cells to do its function. And then androgens in turn stimulate the process of spermatogenesis, a two-step process where LH stimulates Leydig cells produce androgens stimulate spermatogenesis. And then the use of the FSH, follicular stimulating hormone. Follicular stimulation hormone acts on the Sertoli cells and with secretion of some factors which help in the process of spermiogenesis. Basically, this FSH acts on the Sertoli cells and stimulate the secretion of factors FSH, LH, Leydig cells. Some factors which help in the process of spermiogenesis. Now the next topic is examine the structure of a sperm. It is a microscopic structure composed of a head, neck, a middle piece, and a tail. A plasma membrane envelopes the whole body of the sperm. The sperm head contains an elongated haploid nucleus. The anterior portion, which is covered by a haploid structure called an acrosome. The acrosome is filled with enzymes that are called the lytic enzyme, which bears the acrosin and some other enzymes that help fertilization of the ovum. The middle piece possesses numerous mitochondria, which produce energy for movement of tail that facilitates sperm motility essential for fertilization. A human male ejaculates about 200 to 300 million sperms during a coitus, of which for a normal fertility, at least 60% of the sperms must have normal shape and size. At least 40% of them must show vigorous motility. Basically, of a human male ejaculates about 200 to 300 million sperms during a coitus, and of which of these 200 to 300 million sperms, at least 60% of the sperms should have a normal shape and size, and at least 40% of them should have vigorous motility. Unless that is the case, the chances of fertility is less. This is the threshold for fertility for the male. Sperms released from these seminiferous tubules are transported by the accessory ducts. Secretions of epididymis, vas deferens, seminal vesicle, and prostate are essential for maturation and motility of the sperm. Basically, a point to be remembered. Sperms released from the seminiferous tubules are transported by the accessory ducts, and secretions of epididymis, an accessory duct, and vas deferens, an accessory duct, and seminal vesicle, an accessory gland, and prostate are essential. Prostate is also an unpaired accessory gland, and these are essential for maturation and motility of the sperm. Even here, even though the epididymis and vas deferens are not actually accessory glands, they do have some secretions. In such a case, it is a point to remember. The seminal plasma, along with the sperms, constitute the semen. The functions of the male accessory ducts and glands are maintained by the testicular hormones called as androgens. Now, the next process of the oogenesis, the formation of an ovum or the egg, the process of formation of a mature female gamete is called as the oogenesis, which is markedly different from spermatogenesis. Basically, formation of a mature female gamete is what we call as an oogenesis, and it is very different from the spermatogenesis. Oogenesis is initiated during the embryonic development stage when a couple of million gamete mother cells are formed within each fetal ovary. We see that oogenesis initiated during the embryonic development stage when the female baby is inside the mother's womb itself. In the embryonic stage itself, the oogenesis will start. Oogenesis is initiated during the embryonic stage or development stage when a couple, here they are talking about 2 to 3 or many millions of gamete mother cells, which is the oogonia, are formed within each fetal ovary. Basically, oogonia, millions of oogonia, which are the gamete mother cells, are formed with each fetal ovary. Approximately 2 million mother cells, gamete mother cells, which are oogonia, are formed within each fetal ovary, meaning 4 million oogonia in both of the ovaries combined. No more oogonia are formed and added after birth. This 1 million, or 4 million in both ovaries combined, are the final number of oogonia possible, and these no more oogonia are added or formed after birth. After birth, it doesn't happen. Only during the embryonic stages happens. These cells start division and enter into the prophase 1 of the meiotic division and get temporarily arrested in that stage. Basically, in the embryonic stage itself, the cells undergo division and enter into the prophase 1 of the meiotic division, and at this stage, the meiotic division, they get temporarily arrested at this stage of the at the prophase 1, and there is a gap from the embryonic stage from birth till puberty. This is a temporary gap where the cells, the oogonia cells, which started division from meiosis 1 and stopped at the prophase 1, are called as the primary oocytes. Till puberty, the meiosis won't continue. Each primary oocyte then gets surrounded by a layer of granulosa cells and is called the primary follicle. A large number of these follicles degenerate during the phase from birth to puberty. At birth, there were a total of 4 million cells, but at puberty, there will only be 60 to 80,000 primary follicles. That is in ovary. Total 120 to 160,000 in both the ovaries. The primary follicles get surrounded by more layers of granulosal cells and a new theca and are called as secondary follicles. Basically, primary follicle was formed when the primary oocyte was surrounded by granulosal cells, and if more number of these granulosal cells are formed and they get separated by a new theca, these are called as secondary follicles. The secondary follicle soon transforms into a tertiary follicle, which is characterized by a fluid-filled cavity called an antrum. Basically, the secondary follicle again transforms into a tertiary follicle, this is characterized by a fluid-filled cavity called as the antrum. The theca layer is organized into an inner theca interna and outer theca externa. We saw how the ovary and stroma was divided into the peripheral cortex and the inner medulla. Here as well, it is somewhat like that, where there is the interna and outer theca externa, theca interna and theca externa. Here, it is important to draw your attention that it is at this stage that the primary oocyte within the tertiary follicle grows in size and completes its first meiotic division. When it is a tertiary follicle, at this stage itself, the primary follicle, which is undergoing the meiosis 1 division, will complete its meiosis 1 cycle and become a secondary follicle. And this is not an equal division. I'm not talking about the number of chromosome number. I'm talking about the cell sizes here. When meiosis 1, 2 haploid cells should be found, and at meiosis 2, 4 haploid cells should be found. But in the case of an ovum or the oocyte, the primary oocyte divides meiotically in the meiosis 1 to produce only one gamete and a polar body. This is why it is called as an unequal division, resulting in the formation of a large haploid secondary oocyte and a tiny first polar body. Let's see the diagram first. The diagrammatic sectional view of an ovary. There's the ovary that contains the blood vessels. This is the primary follicle. This is the tertiary this is the secondary follicle. This and this is the tertiary follicle, and the final one is the Graafian follicle. When the Graafian follicle ruptures like this, a secondary oocyte, which is a secondary oocyte, this is the characterization of ovulation. And after this broken or torn, ruptured Graafian follicle will develop into a corpus luteum, which is a secretory organ, a temporary secretory organ. The secondary oocyte retains bulk of the nutrition-rich cytoplasm of the primary oocyte. Can you think of the advantage for this? Storing nutrition, basically for later when fertilization happens, division, which will lead to division, rapid division, zygote division, which is called as a cleavage, and this will be helpful. Does the first polar body born out of first meiotic division divide further or degenerate? Most about the polar body is not presently known. That is what they're saying here. At present, we are not very certain about this. The tertiary follicle further changes into a mature follicle or Graafian follicle. Basically, The tertiary follicle here will again mature into the Graafian follicle, and the secondary oocyte forms a new membrane called as the zona pellucida. The secondary follicle, which did have a membrane, will form a new membrane again. This is called the zona pellucida. Surrounding it, the Graafian follicle now ruptures to release the secondary oocyte, the ovum, from the ovary by the process called as the ovulation. Can you identify the major differences between spermatogenesis and oogenesis? Can you? Let's see the diagrammatic representation given below. There is a schematic representation of A, which is the spermatogenesis, and B, which is the oogenesis. This is at puberty. The spermatogenesis starts only at puberty. And the chromosome number for the spermatogonia cells is 46, meaning diploid. Here it undergoes mitosis differentiation to form other spermatogonia cells, and meiotic and primary spermatocytes, some special cells of the spermatogonia are produced when spermatogonia undergoes mitosis, and from primary spermatocytes undergo meiosis, first meiotic division to produce here two secondary spermatocytes. These secondary spermatocytes, as we see here, is 23 in number, and each of these secondary spermatocytes again undergo meiosis 2 to form each two spermatids, which are also diploid. Basically, this is the 4 haploid gametes, and each spermatids again undergo differentiation, which is the spermiogenesis process, to form the main 4 gametes of the sperm or spermatozoa. And in case of the oogenesis, oogonia will undergo mitosis differentiation to form primary oocytes. Primary oocyte is basically primary oocyte. Basically, the first gamete mother cell is the oogonia, and the second is the primary oocyte. Primary oocyte is formed when the oogonia cells undergo meiosis division, but not completely, partially, in the arrested state at the prophase 1 stage. When they are arrested at the prophase 1 stage, then this is called as the primary oocyte. And this is the birth, childhood to puberty gap. In here, there is no formation here, and the primary oocyte completes first meiotic division after the puberty to form a secondary oocyte, the first polar body, and the secondary oocyte again undergoes meiosis 2 to form an ootid or an ovum. This happens only after when the sperm enters when the sperm enters the ovum, but there is no fertilization at that time. Before fertilization, prior to fertilization, but after sperm entering the ovum will divide, the secondary oocyte will divide to give an ovum and a second polar body. Now let's see the menstrual cycle. Okay, let's see the menstrual cycle. The reproductive cycle in the female primates. The reproductive cycle in the female primates. Example, Primata is an order, and example of the order of Primata, which we studied in the first chapter of 11th class biology, is monkeys, apes, and of course human beings. And this is called the menstrual cycle. The first menstruation begins at puberty and is called as the menarche. The first menstrual, important point to remember, the first menstrual cycle begins at puberty, and the main point we remembered is menarche, which is the start of the menstrual cycle. In human females, menstruation is repeated at an average interval of about 28 to 29 days, and the cycle events start from one menstruation till the next one is called as the menstrual cycle. As the menstruation continues to happen, this menstruation cycle is what we call it. One ovum is released. Ovulation during the important to remember is that one ovum is released, that is ovulation. Release of ovum is called ovulation, and it is released at the middle of the menstrual cycle. During the middle of the menstrual cycle. This is the menstrual cycle presentation of various events, diagrammatic presentation of various events during a menstrual cycle. This is basically the cycle. This is the menstrual cycle diagram. Is the uterine events. Uterine events. This is the days of the menstrual cycle, which is from 20, which is approximately 28 to 29 days. And there is the first 5 days is the menstruation. It's actually not the first 5 days, but menstruation followed by the follicular phase, proliferative phase. Then there is the luteal phase, the secretory phase, and then the next phase. Basically, there are 3 phases in the menstruation cycle. There is the menstruation, follicular or the proliferative phase, the luteal phase, and the next cycle. Here, if we see the middle line, there they draw out a line like this. This is the day of ovulation, which is approximately 14 to 15. 14 to 15. The first is the developing follicle, which at the day 1 is small, then it goes on. This is the mature follicle at the day 13 or so. Then follicle is ovulated. Ovulation happens at the 14th to 15th day. Then on the 14th day of ovulation happens, and corpus luteum developed from the ruptured Graafian follicle. This, if there is no fertilization, the corpus luteum is again degenerated, the regressing corpus luteum. And if we see the hormonal levels, the ovarian hormone levels, at the time of ovulation, estrogen is relatively high. There is a peak of estrogen at the time of ovulation, which is basically, I think, inducing the ovulation. Here, progesterone, during the developing phases of the corpus luteum, we see a rise in the progesterone, in the estrogen as well. This rise here, this peak here is what we call, which we attribute to the corpus luteum, which is due to the corpus luteum. This peak here is due to ovulation, and these 2 peaks here are due to the developing corpus luteum. Corpus luteum secretes hormones, which can be progesterone. And here we see a peak here in case of the pituitary hormones here. Pituitary hormones, we see a peak during the 2 peaks peaks here for the FH and LH hormones before the day of ovulation or before the time of ovulation. This is due to LH and FSH induce ovulation. Just before ovulation, there will be a peak here which will induce the ovulation. Of each menstrual cycle, the major events of the menstrual cycle are shown in figure 2.9, the above figure. Cycle starts with the menstrual phase. When the menstrual flow occurs, it lasts for 3 to 5 days. The menstrual flow results due to breakdown of endometrial lining of the uterus. Basically, the first 5 days is basically the menstrual flow. This is due to the big breakdown of the developed endometrial lining. This is a glandular lining of the uterus and its blood vessels, which form liquid that comes out to the vagina. Menstruation only occurs if the released ovum is not fertilized. Menstruation only occurs when the ovum is not fertilized. There is no fertilization. Lack of menstruation may be indicative of pregnancy. Lack of menstruation may be indicative of pregnancy. This is one way to find out if there is pregnancy. However, it may also be caused due to some other underlying causes like stress, poor health, etc. Menstrual phase is followed by the follicular phase. The follicular phase is called the proliferative phase. Follicle is developing, and during this phase, the primary follicles in the ovary grow to become a fully mature Graafian follicle, and simultaneously, the endometrium of the uterus regenerates through proliferation. The endometrium points to be remembered, the follicle develops and the endometrium layer, which through proliferation, it regenerates. These changes in the ovary and the uterus are induced by the changes in the levels of the pituitary and ovarian hormones. The secretion of gonadotropins, LH and FSH, increases gradually during the follicular phases and stimulate follicular development, as well as secretion of estrogens by the growing follicles. Okay, both LH and FSH attain a peak level in the middle of cycle, about the 14th day, which we saw two peaks like this. Two peaks like that. Rapid secretion of LH leads to the maximum level of during the mid-cycle called the LH surge. Important point remembered, when secretion of LH leads to its maximum level during the middle cycle, it is called the LH surge, and this LH surge induces a rupture of the Graafian follicle and thereby the release of the ovum. This is the ovulation, and the ovulation of the ovulatory phase is followed by a luteal or the secretory phase, during which the remaining parts of the Graafian follicle transform as the corpus luteum, and the corpus luteum secretes large amount of progesterone, which is essential for maintenance of the endometrium. Such an endometrium is necessary for implantation of the fertilized ovum and other events of the pregnancy. During pregnancy, all events of the menstrual cycle stop and there is no menstruation. Point to remember, during pregnancy, all events of the menstrual cycle stop and there is no menstruation. This can indicate if there is no menstruation, it might be due to stress and other problems, but it can also be due to pregnancy. In the absence of fertilization, the corpus luteum degenerates. This causes disintegration of the endometrium, leading to menstruation, marking a new cycle. In human beings, menstrual cycle sees us around 50 years. That is termed as the menopause. The start is menarche. Menopause is the end. Pause. Cyclic menstruation is an indicator of normal reproductive phase and extends between menarche and menopause. Menstrual hygiene. Let's see the extra information they give here. Maintenance of hygiene and sanitation during menstruation is very important. Take bath and clean yourself regularly. Use sanitary napkins or clean homemade pads. change sanitary napkins or homemade pads every 4 to 5 hours. As per the requirement, dispose of the used sanitary napkins, properly wrapping it with a used paper. Do not throw the used napkins in the drain pipe, the toilets, or in the open area. After handling the napkin, wash hands with soap. Now let's see the fertilization and implantation. The next main topic is fertilization and implantation. During coitus, semen is released by the penis into the vagina, which is termed as insemination. The motile sperms swim rapidly, pass through the cervix, enter into the uterus, and finally reach the ampullary region into the fallopian tube. The ovum released by the ovary is also transported to the ampullary region where fertilization takes place. Fertilization can only occur if the ovum and the sperms are transported simultaneously to the ampullary region. A point remembered, fertilization can only occur if the ovum and sperms are transported simultaneously to the ampullary region. If here above, if we see this 14 days the day of ovulation, and during this 14 to 17 days is the 14, 17, 19, basically these days is the days of ovulation, and during this not ovulation, this is the fertile period where there are high chances of the fertilization and implantation. Even if there is fertilization in the later phases, if there is no implantation, there is no pregnancy. So in such a case, for fertilization as well as implantation to happen, it should only happen during the fertile period and during in the ampullary region. This is the reason why not all copulation lead to fertilization and pregnancy. Point to be remembered, the process of fusion of sperm with an ovum is called as fertilization. During fertilization, a sperm comes in contact with the zona pellucida layer of the ovum. Let's see the diagram first. Ovum surrounded by a few sperm. This is the corona radiata layer, cells of the corona radiata. This is the pink color one as the zona pellucida layer, and there is also the pink color is not the zona pellucida. This green color, dark green mesh is the zona pellucida, and the inner pink color layer is the perivitelline space. Is a space, a cavity kind of thing, and this induces changes when the sperm with an ovum is called fertile. During fertilization, a sperm comes in contact with the zona pellucida layer, comes in contact with the zona pellucida layer like this, and induces changes in the membrane that block the entry of additional sperm. Thus, it ensures that only one sperm can fertilize an ovum. The secretions of the acrosome help the sperm enter into the cytoplasm of the ovum to the zona pellucida and the plasma membrane. And the plasma membrane, this induces the completion of the meiotic division, the secondary oocyte. The second meiotic division is also unequal. This entering of the sperm induces the completion of the meiosis division of the secondary oocyte. The second meiotic division is also unequal and results in the formation of a second polar body and a haploid ovum called an oocyte. Soon the haploid nucleus of the sperms and that of the ovum fuse together to form a diploid zygote. How many chromosomes will be there in the zygote? Of course, it will be diploid, so 46 chromosomes will be there. There is a sex determination here. One has to remember that the sex of the baby has been decided at the stage itself. Let us know how. At this stage only sex is determined. At which stage? The fusion or fertilization stage. As you know, the chromosome pattern in the human female is double X, and that in the male is X and Y. Therefore, all the haploid gametes ova produced by the female will have the sex chromosome X, whereas in the male gametes sperms, the sex chromosome could either be X or Y. and 50% chance of sperms carry the X chromosome, while other 50% can carry the Y chromosome. After fusion of the male and female gamete, the zygote would carry either XX or XY depending on whether the sperm carrying X or Y fertilize the ovum. The zygote carrying XX would develop into a female baby and XY would form a male. You will learn about this more in the chromosomal patterns in chapter 5. That is why scientifically it is correct to say that the sex of a baby, gender of a baby is determined by the father and not by the mother. But technically, if we say it is not determined by father or mother, it is determined by the nature, because it's not like the father can actually control the rate of which sperm or choose which sperm or which kind of chromosome will fuse with the ovum. So we cannot say it is actually determined by the father. It is determined by the nature. But if there is a question, of course, follow NCERT. In the mitotic division, the mitotic division, the mitotic division The mitotic division starts as a zygote, moves through the isthmus of the oviduct called the cleavage towards the uterus. The cleavage is not talking about the oviduct region, they're talking about the mitotic division. The mitotic division of the zygote as it moves through the isthmus region of the oviduct is called as cleavage, and it is moving towards the uterus, and it forms 2, 4, 8, 16 daughter cells called as blastomeres. The daughter cells formed are called blastomeres. The embryo has many cells formed. It will be called as an embryo. And the embryo with an 8 to 16 cells is called as a morula. Here if we see, the embryo with 8 to 16 is called 8 to 16 blastomeres is called as morula. And let's see the diagrammatic view here. Seems to be glitching. Let's forget about the diagram, I guess. Wait, it seems this is better. This is the way in ovum. This is at fertilization. This is the 2-cell stage. This is the 4-cell stage. This is the 8 to 12, 16-cell stage called the morula. And a morula develops into a blastocyst, which contains more than 16 to 18, 30 to 64 cells. 8 to 16 blastomeres is called a morula, and the morula continues to divide and transform into a blastocyst as it moves towards into the uterus. This is the blastocyst. This is after morula stage. This is the blastocyst stage. Here's the blastocyst contains cavity. Here is surrounded by the trophoblast cells. Contains the inner mass of cells as well. The blastomeres in the blastocyst are arranged into an outer layer called as trophoblast and an inner group of cells attached to the trophoblast called the inner mass of cells. The trophoblast layer then gets attached to the endometrium and the inner cell mass gets differentiated as the embryo. After attachment, basically the trophoblast cells then get attached to the endometrium and the inner mass of cells gets differentiated as the embryo. Okay, the trophoblast gets attached to the endometrium and the inner mass of cells differentiates as the embryo. After the attachment, the uterine cells divide rapidly and covers the blastocyst. As a result, the blastocyst becomes embedded in the endometrium of the uterus. This is called as the implantation and it leads to pregnancy. Pregnancy and embryonic development. After implantation, finger-like projections appear on the trophoblast called as the chorionic villi, which are surrounded by the uterine tissue and maternal blood. The chorionic villi and uterine tissue become interdigitate with each other and jointly form a structural and functional unit between the developing embryo and maternal body called the placenta. Basically, after implantation, attachment, finger-like projections appear that do not on the endometrium but appear on the trophoblast called as the chorionic villi. Then these chorionic villi are surrounded by the uterine tissue and that also the maternal blood and the chorionic villi and the uterine tissue becomes interdigited, forming digits, finger-like projections with each other and jointly form a structural and functional unit developing embryo and maternal body, and this interdigited structural and functional unit between the fetus and the mother is called as the placenta. Statement to be remembered. The placenta facilitate to supply the oxygen and nutrients to the embryo and also removal of the carbon dioxide and excretory waste materials produced by the embryo. Function of the placenta they are telling here, and the function of the placenta is to facilitate, that is, supply of oxygen, supply of nutrients to the embryo and removal of carbon dioxide, excretory waste material produced by the embryo. The placenta is connected to the embryo through an umbilical cord which helps in the transport of substances to and from the embryo. Placenta also acts as a endocrine tissue and produces several hormones like human chorionic gonadotropins, human placental lactogen, estrogens, progesterones, etc. Basically, placenta also acts as a endocrine tissue and it produces several hormones like the HCG, human chorionic gonadotropin, human placental lactogen, estrogens, progesterones, etc. In the later phase, a hormone called in the later phase of pregnancy, in the later phase of pregnancy, in the later phase of pregnancy, a hormone called as relaxin. Relaxin is secreted by the ovary. Let us remember that HCG, HPL, and relaxin are produced in the woman only during pregnancy. In addition, during pregnancy, the levels of the other hormones like estrogens, progesterones, cortisol, prolactin, thyroxine, etc. are increased several folds in the maternal blood. Hormones like cortisol, prolactin, thyroxine, etc. Thyroxine produced by thyroid glands, prolactin produced by the pituitary gland, cortisol produced by the adrenal glands is increased to several folds, several times the normal amount in the mother's blood or the maternal blood. Increased production of hormones is essential for supporting the fetal growth. metabolic changes in the mother and the maintenance of the pregnancy. Supporting this is due to support the fetal growth, basically the baby's growth, the metabolic changes in the mother, and the maintenance of pregnancy. This is the diagram here showing the embryonic development of the human fetus within the uterus. This is the plug of the mucus in cervix. This is the embryo. This is the yolk sac. This is the main middle here is the embryo, the yolk sac here. The cavity of the uterus, this is a red color here. This is the placental villi or placental walls, the umbilical cord here, and the extra vessels. Immediately after implantation, the inner walls, inner cell mass, embryo, that is the inner cell mass, which is the embryo, differentiates into an outer layer called as the ectoderm, an inner layer called as the endoderm, and a mesoderm soon appears between the ectoderm and endoderm. Basically, after the implantation, the embryo, the inner mass of cells, will differentiate into an outer layer. First is an outer layer, then is an inner layer, then a mesoderm is the middle layer appears. These three layers give rise to all tissues, organs in adults. These three dermal layers give rise to all tissues, organs in adults. Basically, why tissues and organs are in bracket is because similar cells working together form a tissue. Similar cells working together for the same function is called a tissue, and similar tissues working together is called an organ. It needs to be mentioned, and some organisms of the animal kingdom also have germ layers, but these are called germ layers, the ecto, endo, and mesoderm, and they are to be remembered and are in other animals as well, where tissues are the only ones. Here, it needs to be mentioned that the inner mass of cells contains certain cells called stem cells, which have the potency to give rise to all tissues and organs. This inner mass of cells contains cells called as the stem cells, and which have the potency to give rise to all tissues and organs. What are the major features of embryonic development at various months of pregnancy? Important thing to be remembered, the human pregnancy lasts for 8 months. Easy to remember. Do you know for how many months pregnancy lasts in dogs, elephants, cats? I don't. Find out in humans. After 1 month of pregnancy, the embryo's heart is formed. Basically, after the first month of pregnancy, the embryo's heart, which is the embryonic heart, is formed. The first sign of growing fetus may be noticed by listening to the heart sound carefully through the stethoscope. By the end of the second month of pregnancy, the fetus develops limbs and digits. By the end of 12 weeks, the first trimester, the pregnancy is divided into 9 months, 3 trimesters, or 36 weeks. Basically, 3 months or 12 weeks is referred to as the first trimester, and 24 weeks and 6 months is referred to as the second trimester, and 36 weeks and or 9 months referred to as the third trimester, which is the end of the pregnancy. Most of the major organ systems are formed. For example, at the first month, the embryonic heart is formed. The second month, all the limbs, digits are developed. And the third month, most of the major organ systems are formed. For example, the limbs and external genital organs are well developed. The external genitalia or genital organs, and the limbs, the digits, these are all developed. The first moments of the fetus, point to remember, the first moment of the fetus and the appearance of hair on the head are usually observed during the 5th month of pregnancy. Basically, on the 5th month of pregnancy, we see hair on the head of the baby, basically, and there is also first movement of the fetus. By the end of about 24 weeks, end of the second trimester, the body is covered with fine hair, eyelids separate, and eyelashes are formed. By the end of the 9 months of pregnancy, the fetus is fully developed and is ready for delivery. Now, I think one of the last topics is one of the last topics or last topic is the last topic, which is one page here, which is basically parturition and lactation. The average duration of human pregnancy is about 9 months. It is called the gestation period. Vigorous contraction of the uterus at the end of pregnancy causes expulsion delivery of the fetus. Basically, the average pregnancy is about 9 months and is called the gestation period or the pregnancy period, and the vigorous contraction of the uterus at the end of pregnancy, contraction of the uterus or the myometrial layer, smooth muscles layer, and this is called this vigorous contraction is called the expulsion or the fetal ejection reflex. This process of the delivery of the baby or fetus is called as the parturition. Parturition is induced by a complex neuroendocrine mechanism. Basically, important to remember, the signals for parturition originate from the fully developed fetus in the placenta, which induce mild uterine contraction called the fetal ejection reflex. Basically, the signals for parturition, there's also a very important point to remember. The signals for parturition, the first signal is originating from the fully developed fetus, and after these signals are originated from the fetus itself, and the placenta, from the signals, the placenta will induce mild uterine contraction. This mild uterine contraction is what we call as the fetal ejection reflex. This triggers release of oxytocin from the maternal pituitary. Basically, the signals from the fetus will induce mild contractions in the uterus, and this contraction is called as a fetal ejection reflex, and this will lead to triggers the release of oxytocin, is contraction enzyme, basically, which is this oxytocin is released from the maternal pituitary. Oxytocin acts on the uterine muscle and causes stronger uterine contraction. This oxytocin acts on the uterine muscle and this causes stronger uterine contraction, which in turn stimulates further secretion of oxytocin. This stimulates further secretion of the oxytocin. Stronger the uterine contraction, stronger the secretion of oxytocin. This fetal ejection reflex is milder contraction. This milder contraction induces stronger contraction, and this stronger contraction increases the secretion of oxytocin even more, and even stronger contractions follow. The stimulatory reflex between the uterine contraction and the oxytocin release secretion continues, resulting in stronger and stronger contraction. This leads to expulsion of the baby out of the uterus to the birth canal, parturition soon after the infant is delivered. The placenta is also expelled out of the uterus. What do you think the doctors inject to induce delivery? It is oxytocin, right? The mammary glands of the female undergo differentiation. Basically, why would if the fetal ejection reflex is working fine, then there is no need to induce the delivery. This can only happen when times like when the baby is complete, the fetus is completely developed, but it is not sending signals when the doctor thinks like that, or when there is a pre-planned induce, like the family planned for such a thing, when the baby is fully developed, the fetus is completely developed, or such a time when little amount of oxytocin, little amount of contraction is released, that will register as a fetal ejection reflex, and it will release the oxytocin secretion and increase the delivery. The mammary glands of the female undergo differentiation during pregnancy and start producing milk towards the end of pregnancy by the process called as lactation. This helps the mother in feeding the newborn. The milk produced during the initial days of lactation is called as the colostrum, which contains several antibodies, absolutely essential to develop resistance for the newborn babies. Breastfeeding during initial period of the infant growth is recommended by the doctors for bringing up a healthy baby. Summary, which is of approximately 1 page. Humans are sexually reproducing, and viviparous the first line of the chapter. And the summary, the male reproductive system is composed of a pair of testes, the male sex axariducts, and the accessory glands and external genitalia. Each testis has about 250 compartments called as testicular lobules, and each lobule contains 1 to 3 highly coiled seminiferous tubules. Each seminiferous tubules is lined by spermatogonia and Sertoli cells. The spermatogonia undergo meiotic division leading to sperm formation, while Sertoli cells provide nutrition to the dividing germ cells. The Leydig cells outside the seminiferous tubules synthesize and secrete testicular hormones called as androgens, and the male external genitalia is called the penis, and the female reproductive system consists of the pair of ovaries, a pair of oviducts, a uterus. Just a minute. Okay, let's continue. The female reproductive system consists of a pair of ovaries, a pair of oviducts, a uterus, a vagina, external genitalia, a pair of mammary glands. The ovaries produce the female gamete, ovum, and some steroid hormones, ovarian follicles in a different stages of development are embedded into the stroma. The oviducts, uterus, and vagina are female accessory ducts. The uterus has 3 layers, namely peri, myo, and endometrium. The female external genitalia includes mons pubis, labia majora, labia minora, hymen, and clitoris. The mammary glands are one of the female secondary and sexual characters. Spermatogenesis results in the formation of sperms that are transported by the male sex or accessory ducts. A normal human sperm is composed of a head, neck, a middle, teeth, and a tail. The process of the formation of mature female gamete is called as the oogenesis. The reproductive cycle of the female primates is called as the menstrual cycle. Menstrual cycle only starts after attaining sexual maturation, puberty. During ovulation, only one ovum is released per menstrual cycle. The cyclical changes in the ovary and the uterus during menstrual cycle are induced by changes in the level of pituitary and ovarian hormones. And after coitus, sperms are transported into the ampulla, where the sperm fertilizes the ovum, leading to formation of a diploid zygote. The presence of X or Y chromosome in the sperm determines the sex of the embryo. The zygote undergoes repeated mitotic division to form a blastocyst, which is implanted in the uterus, resulting in pregnancy. After 9 months of pregnancy, the fully developed fetus is ready for delivery. The process of childbirth is called as parturition, which is induced by a complex neuroendocrine mechanism involving cortisol, estrogens, and oxytocin. Mammary glands differentiate during pregnancy. Point to remember: the mammary glands differentiate during pregnancy and secrete milk after childbirth. The newborn baby is fed milk by the mother. Lactation during the initial few months of the baby growth. With this we have completed first revision of human reproduction. First revision of human reproduction via NCERT. Completed. See you in my next video, and